Last week, we talked about pangenomics, which is a comparative genomics approach that enables us to put together multiple genomes and try to make sense of them given their entire gene content. And we discussed the basic principles behind this strategy, how we identify open reading frames, get the genes out of these genomes, and work with translated sequences to blast them to each other and find associations and describe them as a network from which we find out what genes come together across genomes or within the same genome as clusters, and describe these gene clusters and their characteristics to infer associations between genomes as well as gene clusters. So last week we talked about this concept and used the example of this Prochlorococcus pangenome that was generated from 31 genomes and had a couple of thousand, few thousand gene clusters. And we talked about what do we learn when we take a look at a pangenome like this, and what kind of hypotheses we can develop and follow up upon. This week, we are going to extend this concept and discuss metapangenomics. Metapangenomics is a strategy to co-investigate metagenomes and pangenomes. The schedule of this week is relatively simple, and I'd like to start with the first one quickly, the shortcomings of pangenomes. So pangenomes are excellent, but let's talk about what we are missing with pangenomics. As we discussed during the discussion of pangenomics, one of the holy grails of microbiology is to identify genetic determinants of microbial phenotypes. And we had started with the idea that we need many more genomes. That's the holy grail, creating a genomic understanding of naturally occurring microbial life, even for those populations that we have not yet been able to cultivate. But then, as we talked about it, there are many genomes now. And the availability of many genomes enables us to do new things, such as putting together a bunch of genomes and trying to identify genetic determinants of whatever we're interested in. In fact, this particular pangenome was very useful for that, because if you remember, we talked about the core gene clusters, but besides the core gene clusters that were shared across all genomes that belong to this taxon, we identified high core gene clusters that are core to genomes that survive in high light conditions versus those that survive in low light conditions. And we spoke about the fact that these gene clusters are likely those that can help us understand what is the genetic basis of survival in these different environments within a close-related group of genomes, which reduces the complexity of the problem dramatically through the pangenome. But there is one problem here, a significant problem that we will try to address this week. And the problem is, actually goes back to the most fundamental things about microbiology and cultivation. When we culture an organism from a given environment, we do not necessarily know whether that particular organism is a good representative of all the members that belong to a single population. As you can imagine, a given natural habitat is not made up of monoclonal copies of the same genome over and over again when we're thinking about a single population. For instance, even if you're talking about the human gut or surface ocean or soils or any natural habitats, we'll have a diversity at different levels of resolution. We will have diversity at the level of phyla, so there will be different phyla in a given environment, bacterial, archaeal phyla. But when you look at when you go down and down in the resolution, for instance, from phyla, you go down all the way to species, you will find out there are many different species, but even within a species, the genomes that belong to the members of that species will not be identical to each other. And so when we try to cultivate a member of the species, we may end up cultivating one organism that is not necessarily reflective of the genomes of every other member of this taxon in the environment. And my analogy to that had been, if you recall, if aliens were to capture one human being to talk about the human species, not every accessory this human brings to their ship is going to be representative of all the accessories that are commonly shared across humans, or those accessories that are critical for our survival. So the example was, and I'd like to use again, is that we all have limbs and legs, most of us do, and those are key determinants of our fitness under certain environmental stresses. Like if you have to run, you need to, you may realize that having legs is really important, or and so on. But having long or short hair or having pairs of glasses may not necessarily be as important under all circumstances. So the question here I'd like to raise is, when you cultivate a human being from Earth, or when you cultivate a Prochlorococcus organism from a surface ocean sample, with all the genetic potential in these genomes, how can you determine which ones are the pair of glasses, or which ones are the pair of hands or legs? So you may look at this figure and say, well, we see that here. We see that when we cultivate many of those organisms, there's a core section that describes all the limbs and legs and all the housekeeping things, being able to synthesize amino acids or proteins, etc. And clearly, these are those parts of these genomes that are not necessarily essential. But it's a little more subtle than that in reality, because if you look at this pangenome carefully, you realize the presence of all these singletons. For instance, one could immediately ask the question, well, these singletons, these gene clusters, occur only in one or the other genome. So clearly, they are not necessarily so essential. So how do we make sense of their relevance to the survival of these populations? Or you can turn that question around and say, well, these are core gene clusters, yes, but these are core gene clusters in the sense of the kinds of genomes we have in our collection. Are they really that important as the pangenome suggests in the environment? So these are questions that cannot be answered by looking at pangenomes alone, because these questions, when we start thinking about survival or fitness or distribution across different habitats, pangenomes do not enable us to discuss those things. They give us genomes, they give us the genetic makeup of a given genome, but it does not, a single pangenome does not tell us anything about the ability of these organisms to survive or create very abundant populations. So I hope so far this makes sense. What I'm trying to communicate is pangenomes do provide us an evolutionarily meaningful framework to make sense of genomes given their entire gene pool, which is great. But what we're missing is their ecology, which can be equally important if our purpose is to understand genetic determinants of certain phenotypes, as I mentioned. Luckily, we did discuss some topics that can tell us about the ecology of genomes. If you recall, we talked about metagenomic read recruitment and its ability to help us understand environmental populations given a genomic context or a sequence context. And we had used this toy example where we talked about how we can see, by using a reference context for metagenomic read recruitment from different metagenomes, how we can learn about gene presence absence patterns or relative abundances of a given sequence. Let's say this is a genome. I will not keep saying sequence, but this genome, we can learn about its relative abundance in a given environment compared to other organisms in the same metagenome, or gene presence absence, like these genes are not occurring in this environment, although they are a part of this genome. And then you happen to find these genes present in a given metagenome, even the population itself is not there. So these are some very obvious things we could learn from metagenomic read recruitment, if you recall. And we talked about some basic statistics that could describe this kind of metagenomic read recruitment data, such as coverage and detection. And then we also discussed how these obvious insights include some subtle ones, such as single nucleotide variation that goes even deeper into the diversity of a given population, a given environment, using this reference as a context to describe that environment through metagenomic read recruitment. And so this, I hope, because you are now familiar with this concept, you can imagine how metagenomes could enable us to talk about the ecology at a whole genome level or individual gene level that we use in a pangenome. We can take any genome from a pangenome and try to characterize its gene content across environments. So just to substantiate this cartoon with a real world example, I this time want to show you a single genome across metagenomes, just to give you some insights about how this in reality looks like. This figure, created by Andrew Watson for a paper published by Bunker, Jeff Bunker, and others, to discuss a single organism and its influence on the immune system, human immune system, shows us genes in a single genome and different metagenomes in every layer. So this is a complex figure, but this is not anything different than this actually. So in the next figure, I'm showing you every spork is one of these genes, and every layer is one of these environments. And here you see a lot of those environments, not 3. There are more than 300 different metagenomes at every layer, and these metagenomes are they happen to be coming from individuals who lived in different countries. For instance, this set of metagenomes, there are 150 layers here, are from people that lived in the US. And these layers, for instance, coming from people who live in Tanzania or Fiji or China and so on. So, and every single one of these sporks, I don't know how to call them, items on this display, is a gene from this genome. And unlike in our previous figure, genes in this genome are not organized in this display based on their synteny, but they are clustered based on their read recruitment profiles. That's why genes that are recruiting reads from all metagenomes are coming together like this, even though they may be distributed to different places in the genome. And genes that are not recruiting anything at all or recruiting very little are coming together like this here. So the reason I'm mentioning it, this particular figure, is to highlight a very key critical point that we had been discussing about. This genome was cultivated from a human gut. This is a Ruminococcus navis genome that belongs to species Ruminococcus navis. That is a common member of the human gut microbiome. And so Jeff Bunker and his colleagues had been studying this genome to characterize its immune profile. How does it influence the host immune system? Most critically, based on 2 genes that were encoded by this genome. So the question is, of course, you start asking yourself at some point, is whether those genes that seem to be most critical for the phenotype that we observe are common to all Ruminococcus navis populations that are out there. So when you cultivate a single organism, if you are unable to say whether all genes are relevant to a given population, then this question becomes very important to investigate. And for instance, by doing this analysis, Andrea was able to show the distribution of this population across healthy individuals and doing it at the gene level. And I don't want to take anything away from Andrea's effort, but this is not necessarily a difficult thing to do. You all can do that. Tools for doing this are there, and it is one of the most standard things we do in genome-resolved metagenomics, read recruitment and profiling them at a high resolution. So when you look at this, you immediately recognize multiple things, and perhaps this even makes you feel like as if you are looking at a pangenome, right? There are certain parts. If you compare it to the Prochlorococcus pangenome in your mind, you can say, oh, this is the core, and these are accessory gene clusters that are perhaps not necessarily specific to one country or the other. But unlike pangenomes, there's something subtle here. And that subtlety comes from the fact that the genome, this Ruminococcus population, this genome belongs to, that represents a member from, is not necessarily occurring in every metagenome. And for instance, when you focus on Tanzania, you can see that there is no signal because the presence of a data point indicates coverage of a gene. So if the gene is present, then in the environment, if it's recruiting reads from a given metagenome, then there's a data point here. The lack of data points in Tanzania metagenomes indicates us that the population from which we cultivated this Ruminococcus navis is not in those gut environments. So you immediately recognize which metagenomes actually have this organism. And when you know which metagenomes do not contain a population that resolves to this, you immediately recognize that these genes here that continue to recruit reads are actually those we described here. Let me go back quickly, like these ones. They are there even when the population is not, which means these are not necessarily specific to our taxon, but perhaps they occur in a much broader distribution across different microbial organisms. And then you recognize parts like this. These are likely hypervariable genomic islands that represent genes that are not necessarily essential for this population, but they are occasionally occurring in some genomes and not others, and so on. So these are like accessory genes. They would have likely been accessory genes in a pangenome if we had multiple of this genome. But we are now looking at a single genome across multiple metagenomes. And then you recognize this section, for instance, here, that kind of a smooth signal going all the way here. And those are the genes that are present in a given metagenome when the population is present, and often is a single copy by looking at coverage statistics here. So you can immediately recognize for a single genome by using read recruitment these essential properties of genes. So this is something very straightforward to do, and I hope you can keep this in mind when we're discussing metapangenomics in a second. And there are different properties to this display, such as being able to talk about how many reads that are recruited and the detection of this genome in different metagenomes. And the most critical point we were able to infer from this is that these two genes that were key for this particular study that the group had been studying in the lab was not occurring here, for instance, but was occurring in the core part of the genome. So whatever conclusions one may have about the function of these genes could be could be translated to a more generalized understanding of this taxon and its involvement in the human microbiota. I hope so far this makes sense. I have not, the only thing I've been trying to do was to show you how metagenomic read recruitment in real world settings can tell us about individual genes in a genome and its distribution and its ecology across metagenomes. So keeping that in mind, let's talk a little bit about how do we, let's talk about how do we even compute a metapangenome. This is going to be somewhat similar to our metagenomic, pangenomic example. We will start from there, actually. So just like if you recall, in the pangenomic example, we had a bunch of genomes, and we managed to turn them into this kind of data where genomes and gene clusters are identified, and relationships between one another is shown on the display. So now what I'm going to try to do here with you is to expand this display and turn it into a metapangenome. So what does it even, how does it even occur? If you remember how we took one genome in the example of Ruminococcus navis and run it across multiple metagenomes and learn about its properties at the gene level using metagenomic read recruitment, we can take one of these genomes, there are 4 of them as you can see, and ask the question, if we have any metagenomes, what does the read recruitment of this particular genome in this metagenome looks like? And let's say this is how the read recruitment turns out to be. So this way we learn that this genome there's a population in this environment that we can access through this genome, and we can talk about its relative abundance here. So then we can add this new column to this figure for this particular genome, genome 1, in metagenome 1, and add the average coverage of this genome in this environment to communicate its abundance. And we can add this particular bar here. And now we can go on and use the read recruitment statistics for genome 2, let's say it's only this much, to add the relevant information for genome 2 in this display. So we are essentially building more information about these genomes in corresponding metagenomes that are shown here. And let's say this genome 3 in metagenome 1 is covered like this. And so we represent information there. And let's say genome 4 looks like this. And so we represent this information here. And now we have characterized all our 4 genomes in metagenome 1. So we add this column here. One could do the same thing for another metagenome or another metagenome if they have data. So for instance, now I essentially characterized all these genomes in all metagenomes I had access to, and I learned the coverage statistics so I can actually show how the relative abundances of these genomes compare to their organization here. Just to revisit this idea, we had associated these genomes to one another based on gene cluster distribution patterns. How gene clusters are shared across these genomes had enabled us to talk about how they relate to one another. And we had discussed about the fact that genome 1 and 3 are most similar to each other versus genome 2 and 4 because of this gene cluster 7 here. Now what we're seeing is this. So the gene pool of these genomes enable us to infer some associations between them. And now when we discard that information and go to the environment and ask the question how they are distributed across different environments, we are seeing that that organization by gene clusters actually reflected on their relative abundances in different environments. So what does this tell you? This tells you that whatever the determinants of this organization at the gene pool level is also reflective of the ecology. So we immediately are able to link this information to this information, because as you can imagine, this could have been completely random, in which case we would have concluded that we are not learning anything from the pangenome that could be translated to their ecology. Okay, so I hope so far this makes sense. Essentially, these data points I want to reiterate are coming from the summaries of these read recruitment statistics at the whole genome level. I hope that's clear. There is one more thing we can do, of course, and that thing is to start focusing on individual genes. Because as you can see, not every single gene is created equal in this genome. And there are certain properties that are captured by the abundance of this genome across these metagenomes that may not necessarily be uniformly identical across all genes. For instance, in metagenome 3, we know that this population does not occur because there is no read recruitment except this one gene, which happens to be a gene that recruits non-specific reads, perhaps coming from other populations. And I hope this part makes sense, because this is key for most of the things we're going to be talking about. Yet, if you were to look at the genome 1 across metagenomes here, you can see some small amount of read recruitment as if the population is present there. You can see how this could be misleading if there are many genes that are conserved in a given environment, even when the population is not there. So to prevent from those kinds of misunderstandings, what we can do is to characterize individual genes. For instance, I would like to ask this question and explain what that means. Which genes in this genome are present in metagenomes in which the population itself is present? So this is a mouthful, but what I'm trying to say is, by looking at this, I know that this genome represents populations in metagenome 1 and metagenome 2, but not in metagenome 3. So now I am asking, which genes in this genome follow this pattern? Individual genes, which ones of those are occurring in metagenome 1 and 2, but not in 3? Because those are the genes that follow the distribution profiles of the population itself. So as you can see, A fits to the criterion very well, and B does, and C does, but not D. D is doing its own thing. So D does not care about the distribution of this genome across these environments. But E is doing it very well. So there are multiple genes in this display that behaves like D, even in this cartoon. For instance, the other two are B and C that do not necessarily follow the distribution profiles of the genome. So now I want to turn this into an idea and talk about that these genes, the green ones, represent environmental core genes for this population. And this D represents environmental accessory genes. Because just because D is not found in these metagenomes does not mean that D is not a part of this genome. It still is a part of this genome, but it perhaps is not as important for the survival of populations in these metagenomes as the other ones are. I hope that makes sense. So D is still a real gene, but has a different property compared to C. And that property makes that an environmental accessory gene rather than being an environmental core gene. And the same goes for B here and C. So now basically what I'm doing is that I'm introducing a new concept, environmental core and environmental accessory nature of genes. Just like the way we had core and accessory genes in our pangenome, as you can see from this cartoon example, we can talk about environmental core and environmental accessory nature of individual genes. How does one, what is it that we should do to summarize this information in this context? So, okay, with this, I'm going to add one more column, one more row to our pangenome. We added our columns for metagenomes. Now we are adding a new row to talk about each gene cluster and whether they are environmental core or environmental accessory and to what extent they are. For instance, how could one identify the nature of genes in gene cluster 4? So for this, we need to go back to the pangenome and learn that gene cluster 4 had these genes in it. If you remember our network analysis to identify gene clusters, we know exactly which genes contribute to a given gene cluster. We knew when we did our blasting and resolving the associations between genes that genome 1C, genome 2D, genome 3B, and genome 4B, all these genes came together because they were very, very similar to each other and ended up forming this gene cluster 4, which is shown here. So because we know gene-genome associations, we can ask the question, which genes in what genomes correspond to this gene cluster? So obviously, genome 1C is a part of this gene cluster, genome 2D is a part of this gene cluster, and we can ask the question, what's the proportion of genes in this gene cluster that seem to be environmental core versus environmental accessory? And for instance, in this particular case, every single gene in this gene cluster is environmental core. So they are all green, 100% green. And for instance, for the next one, we can go for gene cluster 8, gene cluster 1, and so on. So all these gene clusters are filled with genes that are environmental core when you analyze the genomes individually across metagenomes. But how about gene cluster 6, which contains a single gene from genome 1D? So genome 1D is an environmental accessory gene. So 100% of this gene cluster is going to be essentially red. And then the last one we have is gene cluster 5, which contains these 3 genes in it, and 2 of them are coming from genome 2. When you try to summarize that information, you will realize that, in fact, one of them is environmental core. So it will be 33% core and 33% accessory. So when you look at this, you realize 2 things. By linking environmental distribution patterns through metagenomics and pangenomes, we can learn about the ecology of and distribution patterns of genomes across environments and the nature of gene clusters corresponding to based on read recruitment results. So we can find out and ask the question, if a given gene cluster is core to the pangenome, is it also core to the environment? or if a gene cluster is a singleton that is not necessarily key to the pangenome according to what we can learn from pangenomes, but is it found in every environment this population is found. So this can help us recover from the fact that certain sequences can be split into multiple gene clusters due to sequence similarities when they are doing functionally similar things, and perhaps may be critical to the population. So this could enable us to divide singletons into multiple groups that seem to be important and that seem to be conditionally important, perhaps. I hope this is making some sense. Are there any questions? Should we stop, take a break, and discuss those questions? Okay, let's continue then. Either everyone is lost or everyone is on board. I will just assume that we're doing well and continue. I know that this is an advanced concept. This is very difficult to communicate to even audiences that are well versed in pangenomics and metagenomics. But I wanted to bring this up because I think this sheds light on a lot of things that we need to keep in mind when we're looking at pangenomes or studying metagenomes, even if you are not going to be performing metapangenomics on any of the data you have. Okay. Let's talk a little bit about metapangenomics in practice. So I hope the basics of this principle is now somewhat clear to all of you. And what I want to do is to use the example from the last week, the Prochlorococcus pangenome, and turn it into a metapangenome. And if you recall, what you're seeing on this figure is essentially identical to this. So we have our pangenome, we know our gene clusters. As we talked about it before, this figure is like somewhat bent version of this figure. And we have these core gene clusters, we have extra gene clusters and everything. What we want to do now essentially is to extend this figure with this information. So how could one do this? When I have this pangenome, how could I add this layer of information here? Well, essentially, I would need to have access to metagenomes that are commonly including populations that result to organisms in pangenome. So now we're talking about Prochlorococcus, which is a surface ocean microbe that occurs in sunlit ocean. Then we need to find metagenomes that are coming from the same environment to be able to do these read recruitment analyses and not get empty lines for every single metagenome, right? Luckily, we have those metagenomes, and they are coming from Taro Ocean's project. That is a project that it's a multi-institutional project led by a lot of individuals, and essentially they went around the Earth's oceans and collected samples in a very highly resolved fashion. And they generated very deep sequencing of these samples, and they released it for public use. So essentially, there is this resource that you can download metagenomes for all these stations that are shown on this map. Since now we have these metagenomes, we can actually do what we talked about, using every single genome across all these metagenomes and characterizing read recruitment results to create that matrix of distribution patterns for our genomes. Essentially, if you were to do that, I'm not going to go into practical details of this, but this is very doable. When you do that, you can extend this figure with the summary of the distribution patterns of these genomes across metagenomes. So essentially, what I just added here is nothing more than this red part here. It's just more populated because we had many metagenomes, and it has more rows because we have more genomes. But essentially, just to orient you a little bit more to this figure, is that the read recruitment results from this metagenome across all genomes correspond to this row at the very top, And this one is somewhere in the middle. And the read recruitment results of all genomes from this particular metagenome is shown here. So they are organized in such a fashion that they follow the organization of these samples on a map. So I will just focus on this section for a minute, but I want you to appreciate the fact that it doesn't matter what system you are studying. What we're discussing right now is applicable to anything. If you have, let's assume you have a culture from a rhizosphere community, there are metagenomes from the rhizosphere. And luckily, metagenomes are not suffering from biases of amplicon sequencing. You can use read recruitment to truly investigate if your population is occurring beyond taxon names or low resolution identities of short sequences. If you are working with gut environment, if you have cultures from the gut, or if you're interested in a particular genus or species in the gut environment, there are human microbiome project metagenomes that you could download and use. So there are many resources for metagenomes for almost every environment, and there are many genomes that you are generating through by reconstructing them from metagenomes, or by isolating them, or applying single cell strategies, and others generate by using these strategies and create these large databases. So you have the data. If you have a question related to both the evolution and ecology of populations you're studying, actually all of these are applicable to pretty much every system. We're just using ocean as an example. And to further discuss the utility of this approach, I will focus on this part a little bit. So last week we discussed that these clades represent different ecotypes of different populations of Prochlorococcus that are adapted to different environmental conditions. So we talked about the fact that these highlight groups, highlight group 1 and highlight group 2, tend to occur in higher, lower depths of, should I say higher depth or lower depth if I want to go closer to the surface? I'm not sure. But they are closer to the surface. And then these low light clades don't deal very well with high light situations, so they tend to be a little deeper in the ocean. But if you look at this very carefully, we have a lot of genomes from highlight clade 2. The reason all these genomes are clustered into highlight clade 2 is because they are way too similar to each other to describe other subclades. So they're almost identical. They have nearly identical 16S ribosomal RNA genes. They are nearly identical at the average nucleotide identity level when you compare these genomes to each other. And that's why they are coming together like this in the pangenome. So the differences between them are quite minuscule. If you, for instance, if I were to go back to this display just for a second and show you, this is highlight group 2. And if you follow all the gene clusters in this that matches to highlight group 2, you can see that there are not many differences between them. So they are nearly identical at the level of their gene pool too. That's why they are all described as highlight group 2. But when you focus only on highlight group 2 genomes and focus on the data shown here, you realize something interesting. And that is that there is this gradient of the color. So this is, in fact, very critical for multiple reasons. First of all, I want you to think about for a second this dendrogram. The dendrogram that organizes all these columns are created when we focused only on the pangenome. So we did not know anything about these patterns here, right? And therefore, the intensity of the color we observe here did not inform our organization of these genomes. We looked at their gene pool, and then we found out that the highlight group 2 genomes are organizing in a very specific fashion. And we couldn't know why, because pangenome does not tell us why is this happening. So just for a second, if you look at this, the organization here is represented by a very small number of gene clusters, as you can see here. It's not like there are a lot of gene clusters that seem to be enforcing this organization. So they are very similar. Yet, when we do read recruitment, and this is competitive read recruitment by which I mean we allow these genomes to be able to recruit from any given metagenome with equal chances, and only those reads that are matching them to perfectly is going to map to them. But when a short read from an environment matches two different genomes, it will be assigned randomly. This is what we generally call competitive read recruitment, which means all these genomes had equal chances to recruit reads from any given metagenome. If we focus on the very top one, let's say A and W is Atlantic Northwest. It's just a station in Taro Ocean. So if you look at the very top red row, you realize that the read recruitment favored these 4 genomes more than these 3 genomes, for instance. There's a very stark difference between their occurrence, which indicates that at this depth, whatever depth this sample is coming from, this group of genomes are recruiting almost an order of magnitude more reads than this group of genomes. Then you realize something very interesting. When these genomes are nearly identical, how can there be such a difference in their occurrence profiles in the same oceanic region? Then you start realizing, if you discard the ocean part of the story and focus on the concept only, you may have nearly identical genomes differed by only a handful of gene clusters that may have very different ecological properties. If we were to, now thinking about this a little more from a philosophical point of view, perhaps, if we are comfortable feeling that the abundance of a given population in a given environment is a proxy to talk about its fitness in that particular environment, then we could say that among these nearly identical genomes, these group of genomes are more fit in this environment than these group of genomes. I hope that somewhat makes sense, because every time we compete them, like how many of you guys occur here, we end up recruiting 10 times more reads for these genomes. And there are a lot of things to talk about technicalities of doing this and making sure that we are not shooting ourselves in the foot by introducing some sort of bioinformatics bias. But we did talk about these things in other discussions, and unfortunately, I'm not sure how to summarize them very well, so I'm skipping it. But if you trust my word with respect to read recruitment and its characterization, what I'm telling you is that these genomes are recruiting a lot more reads than these in a consistent fashion across all ocean realms we have from the temperate oceans. And this is the important detail that is also confirmed by the organization of these genomes by the pangenome, which means there are actually gene clusters here in this not very well characterized, not so well pronounced regions of the pangenome that can tell us about the likely determinants of fitness for this kind of rain, like how these genomes are doing so well compared to other genomes here. The answer to that, at least the beginning of an answer, is somewhere here. And in fact, when you ask the question to the pangenome, I am going to look at gene clusters between these 4 genomes here and 3 genomes here, and I'll focus on every gene cluster that occurs in all 4 and never these 3, with the assumption that these genomes will have some extra, something that makes them more able to survive in this environment. You find 50 gene clusters in this pangenome. 50 gene clusters that are commonly found in these genomes and never found in these genomes. And only 25 of those gene clusters, out of 50, only 25 of those gene clusters actually have known functions. And those functions include DNA repair, many of the known determinants of, someone doesn't like the content. Some of the very well-known determinants of fitness in a surface ocean setting. And confirming that perhaps those gene clusters enable these guys to compete better. In contrast, 25 of those gene clusters do not describe functions that we recognize, which is very exciting, in my opinion at least, because from 7,000 gene clusters, you can reduce to 25, a handful, if you want to learn something that maybe is important as DNA repair for the survival of populations in the surface ocean environments. So this, I hope this kind of gives you an idea about the power of linking these ecological patterns to our understanding of the entire gene pool. Okay, so we essentially, by doing metagenomic read recruitment per genome and characterizing them like this, we completed this part of this figure. So we have an idea about the overall gene distribution patterns. But I want to remind you that when we were looking at the genome one across 3 metagenomes, we had talked about this little blip here that was so unreal because it was there due to the presence of a single gene that even though the rest of the genome 1 was missing in metagenome 3, right? So then, in fact, you can ask yourselves, how do we know these data points here, each of these cells here are summarized from the entirety of the genome? So you can ask yourselves, how do we know that this signal is not due to the presence of a single genomic island that recruits so many reads? For instance, just to substantiate that point, it could have been the case that these organisms, these 4 that we had been talking about as more fit versions of highlight group 2, may have been infected by a phage that also infects a lot of other populations outside of Prochlorococcus, and all the reads may be coming from that region of the genome. So actually, it could have been the case that these genomes, all of them are identical in their distribution in the environment, but only a single region in these in this group of genomes contribute to their increased relative abundance. It could have been the case. So that is a very important question to keep in mind, and one we can answer actually. And answering that question requires us to focus on individual genomes at the gene level, so we can, in a highly resolved fashion, describe gene distribution patterns, which would tell us whether there are some genes that are disproportionately well covered than others, etc. And it would add this section of this figure by being able to characterize each genome independently and summarizing that information at gene cluster level. So now I'm going to talk about that. I hope we are still together. We're not completely lost at this point. Okay, so let's start with one genome. Essentially, I will take one of these genomes here, only one, and the column that that shows us its distribution patterns. And let's take MIT 9314, and which happens to have a distribution pattern like this across oceans when you summarize the entire information. But what I will do now, I will only show that single genome and its genes across metagenomes. So in this particular case, the genes in this genome are organized based on their synteny. Unlike the previous figure I showed you that Andrea had created, in this particular example, this is the first gene that occurs in the genome. It goes all the way here, and this is the last gene here. So if you ignore this white gap that I introduced to make sure we know where the genes start and end, when you look at the rest of the genes in this genome, you realize that there are interesting patterns happening. For instance, there's a section in this genome here that describes a set of genes that are next to each other in the genome that never occur in these metagenomes, or they don't occur in any level of detection. So basically, we are seeing a genomic island that seem to be absent in most of the members of this population in the environment. Because if you focus on, so every layer is a metagenome. I hope you remember that. And these are different oceanic regions: Indian Ocean North, Indian Ocean South, Pacific Ocean North, and so on. And these presence of near perfect circular signal indicates that this particular genome, with almost all of its genes, is present in that one metagenome, right? So using those metagenomes in which our population is certainly present, we can talk about whether other genes that belong to this genome is present there as well, and across and systematically across every metagenome in which the population is present. And when you do that, you can actually classify these genes into 2 categories: environmental core and environmental accessory. And your eyes already tell you which ones are environmental core, which ones are environmental accessory at this point. but one could write a program to do that and look at those very simple statistical properties of these genes and metagenomes. Say these are environmental accessory genes, for instance, here, and this one too, and that one too. And the rest are environmental core, because we detect those genes in the environment every time we detect the entire population. I hope this concept comes through. So this is only one genome in that pangenome. We could do the same thing by using another genome. For instance, we could characterize, and this is a highlight group 1 genome that occurs in colder temperatures. That's why it looks a little bipolar. If you remember, the organization of these metagenomes was kind of going from the north and temperate oceans and south. So therefore, this is north and temperate oceans and south. So we see that you immediately recognize what people already discovered and described in the literature, that this is a cold temperature-loving version of the highlight group. And here we also see the same pattern. There are some genomic islands that represent hypervariable genes that do not necessarily occur in every environment the population is found, but there are others that are core. So again, we get to describe environmental accessory and environmental core genes at the genome level by using nothing more than read recruitment. And we can continue doing that for each genome. And one can actually describe all the genomes that there are in this pangenome and learn about these characteristics per gene. I hope you recognize that this is identical to what we did in our cartoon example. We are essentially using read recruitment results to classify our genes into 2 categories. So, and again, if you remember the cartoon example we discussed, one can look at each gene cluster and ask the question, what are the proportions of environmental core and environmental accessory genes in each one of them? And then you can, when you do that, you can extend this display with this new layer that shows those proportions. So I am not going to go into the details of the biological findings of this, but I will give you a title paper if you'd like to follow up on that. But essentially, if you look at what we are seeing here is that many of the singletons that are in the pangenome, that are singletons that, because they are not necessarily core to all the genomes, are red. So those that are specific to a single genome tend to be absent in environmental populations, which is kind of confirming what one would expect. And in contrast, when you look at the core section, things that seem to be so essential that they become core gene clusters in the pangenome are commonly found in environmental populations, because there's a lot of green here. So when you look at the green versus red, it's very clear across different sections of the set of gene clusters. And then, for instance, highlight core gene classes seem to have a little more red. Low light core seems to have a little less red, and so on and so forth. There's a lot going on. Of course, one of the most important things is asking if there are gene classes that are core to the pangenome, but largely accessory to the environmental populations. That would have been very interesting. And of course, the devil is always in the details, and there indeed are gene clusters that are behaving so differently than what you would expect. Because now what you are seeing is that there is this core set of gene clusters that have to be present in every genome, even when they're coming from entirely different clades of Prochlorococcus. But when we do read recruitment for individual genomes, we found out that some of those gene clusters that are core to the pangenome are largely accessory to the environment, which brings up a lot of new questions and ways to, of course, investigate, and will depend on the project, but gives us an understanding that we couldn't have had unless we included metagenomics into this pangenomics investigation. And that's described in this paper, these patterns a little more in detail with more hypotheses about why is this even happening. But I want to go back to this and kind of finalize this discussion. So essentially, metapangenomics is a systematical and quantitatively tractable strategy to link the gene pool of a set of genomes to the lifestyle of populations with which they are affiliated. In this particular case, for instance, we used Prochlorococcus genomes and surface ocean metagenomes to create a pangenome for Prochlorococcus, talk about its gene pool and how these genomes relate to one another given their entire gene content, and used surface ocean metagenomes to understand their lifestyles. This strategy exploits the nexus between pangenomics and metagenomics at various levels of resolution. So we can learn about the abundance of populations across metagenomes, and we can talk about gene cluster level accessory or core nature of individual genes. And the beauty is that it is not only applicable to isolate genomes, or it's not only applicable to metagenome assembled genomes. If you have any kind of genome or a combination of these two, and metagenomes in which you expect your populations to occur, you are ready for metapangenomics, or different, even if you're not interested in metapangenomics itself, perhaps different components of it. Maybe you have a single genome and you don't care about pangenomes, and you want to understand if this one genome you have reconstructed from a metagenome or isolated from a culture, whether all genes you are studying are core to the environmental populations that are represented by this genome. You may be interested only that part, and it is also very accessible. Okay, so just to recap what we had been doing during the last 3 weeks, we talked about metagenomic read recruitment, and we learned about how metagenomic read recruitment, when you have multiple metagenomes in a genome, can tell us about these gene distribution patterns that may be essential to understand the ecology and evolution and the gene content of these of genomes we're studying. And then we talked about pangenomics that enabled us to put together multiple genomes that are somewhat closely related, for instance, from the same species or same genus, but even members of the same phylum can come together, and learned about core and accessory nature of genes shared by these genomes. And we used the Prochlorococcus pangenome to substantiate some of those points with a real-world example. And then we extended our pangenomes by combining them with metagenomic read recruitment to talk about metapangenomics, how this display turned into this display. I tried to explain that this week. And then we substantiated that point by using a real example from the real world, a real world example using Prochlorococcus. So this is all I have to say about metapangenomics this week. Now I'm going to start the discussion session in a second, but I want to highlight something interesting here, and that is this dendrogram here, the clustering dendrogram that organizes these genomes, can also be reconstructed using phylogenetics or phylogenomics. So that is actually such an important aspect of everything we're doing. I think it's very timely that next week we're going to be talking about phylogenomics with Mike Lee, and try to understand how do we infer evolutionary relations between microorganisms using core genes that they have that result to gene families that are essential to many of microbial genomes. And then we will have a little discussion then to discuss how phylogenomics connects back to pangenomics, and what can we learn by the combination of the two to do more highly resolved analyses and talk about horizontal gene transfer and so on. So with that, I thank you very much for your time. And as always, I am very thankful for my group for helping me with the science, and also letting me do these things in the expense of my time with them. And I thank those who support our science.